Good morning. It's July 22nd, and this is your daily brief in science. Here's everything you need to know. Researchers have developed a non-specific mRNA cancer vaccine that stimulates a strong immune response, moving beyond traditional approaches that target specific tumor markers or mutations. When combined with PD-1 inhibitors, this vaccine has significantly improved treatment outcomes, sometimes even leading to complete tumor elimination. Dr. Dwayne Mitchell highlighted the potential for creating a broad, off-the-shelf cancer vaccine effective across various cancer types. The promising findings have been published in Nature Biomedical Engineering, marking a major milestone in cancer research. This research signifies a significant step towards a new era in cancer treatment, leveraging innovative mRNA technology. The study underscores the growing promise of mRNA technology in oncology, with calls for further studies to validate these results in human trials. Senior author Dr. Elias Sayor emphasized that this approach offers a novel treatment pathway that could potentially replace or supplement traditional methods like surgery, radiation, and chemotherapy. The vaccine could reduce reliance on invasive treatments, providing a less invasive option for patients. Supported by organizations such as the National Institutes of Health, this research could lead to a universal cancer vaccine that is easy to produce and administer, potentially transforming global cancer care. Building on previous successful trials with personalized mRNA vaccines for glioblastoma, this study broadens the potential application to a wider range of cancers. The research extends earlier work, demonstrating that personalized mRNA vaccines can reprogram the immune system to attack aggressive brain cancers like glioblastoma. Further tests showed that a modified version of the vaccine could activate the immune system independently, achieving beneficial effects and even eliminating tumors in some cases. Nanoviricides Incorporated has developed NV-387, an antiviral drug that has demonstrated promising results in animal studies against measles. In these studies, the drug increased survival time by 130% without causing toxicity. The research used specially modified mice expressing human proteins necessary for measles infection, highlighting the innovative approach of the study. NV-387 works by mimicking cell surface features that viruses like measles require for infection, effectively binding to and destroying the virus before it can infect cells. The drug is now moving into Phase 2 human clinical trials. In addition to measles, the company is exploring its broader antiviral technology to target other viruses such as RSV, COVID-19, and influenza. Nanoviricides holds exclusive licenses for various antiviral technologies, but emphasizes that drug development is a lengthy and capital-intensive process with no guarantee of success. The need for effective measles treatments is urgent due to declining vaccination rates and rising infection cases globally. Current vaccination efforts are insufficient, as some vaccinated individuals can still contract the disease. Achieving at least 95% vaccination coverage is essential to prevent outbreaks, but vaccine hesitancy and declining rates threaten this goal. NV-387 functions as a cell decoy by mimicking cell surface features, binding to viruses that rely on sulfated proteoglycans for infection and destroying them before infection occurs. This mechanism targets 90 to 95% of human pathogenic viruses that depend on similar binding features, making NV-387 a broad-spectrum antiviral candidate. The company aims to advance NV-387 into Phase 2 trials, although the timeline for an investigational new drug application remains uncertain. Despite these promising results, Nanoviricides notes that drug development is a lengthy, costly process, with no guarantee of FDA approval or successful outcomes. The success of NV-387 against measles builds on previous achievements against RSV, leveraging the common use of heparan sulfate proteoglycans by these viruses for initial cell attachment. Researchers have made a notable advance in Alzheimer's research by combining two existing cancer drugs, letrozole and irinotecan, which has shown promising results in mouse models. These drugs significantly reduced brain degeneration and improved memory performance in the study. The discovery was made through the use of advanced computational tools that analyze data from various studies to identify drugs capable of reversing gene expression changes associated with Alzheimer's. Further analysis of electronic medical records indicated that patients taking some of these drugs for other conditions had a lower risk of developing the disease. The findings were published in the journal Cell on July 21, 2025, with support from the National Institutes of Health and the National Science Foundation. Alzheimer's disease affects approximately 7 million people in the United States, and current treatments mainly manage symptoms rather than alter disease progression. Despite the positive results in preclinical models, side effects such as hot flashes, severe diarrhea, nausea, and vomiting raise concerns about tolerability in humans. The study's co-senior author, Yadong Huang, highlighted that the complex genetic alterations in Alzheimer's make traditional single-target drug development difficult, which is why multi-target strategies are gaining attention. Unlike conventional treatments, this combination therapy aims to address multiple brain cell types by reversing gene disruptions, potentially offering a more effective approach. The research emphasizes a shift toward multi-target strategies to overcome the high failure rate of single-drug approaches in Alzheimer's. However, the researchers stress that human clinical trials are necessary to confirm safety and efficacy before considering these drugs for widespread use. The team is planning to move into clinical trials soon to evaluate the safety and effectiveness of this drug combination in patients. Astronomers have identified a dim companion star to Betelgeuse, the prominent red supergiant in Orion, using the Gemini North Telescope in Hawaii. This companion, nicknamed Beetle Buddy, has an estimated mass of about 1.5 times that of the Sun and orbits Betelgeuse at roughly 4 times the Earth-Sun distance. It is still in the early stages of stellar development, not yet having begun hydrogen fusion, and is classified as a pre-main sequence star still accreting material. Betelgeuse, by contrast, is nearing the end of its life and is a well-known variable star with brightness fluctuations and recent dimming events. The gravitational influence of Betelgeuse suggests it could potentially consume this companion within the next 10,000 years. The companion star is less than 1 solar mass, about 20 times less massive than Betelgeuse, and approximately 1 million times fainter. Previous observations with NASA's Hubble Space Telescope and Chandra X-ray Observatory could not detect this star, but the new discovery clarifies that Betelgeuse's 2019 to 2020 great dimming was caused by a dust cloud, not an imminent supernova. The findings, published in the Astrophysical Journal, improve understanding of red supergiant variability and their dynamics, especially through spectroscopic binary observations. Further observations are planned for November 2027, when the companion will be at its maximum separation from Betelgeuse, allowing for more detailed study while it is within observable range from Earth. A recent study using EEG technology has uncovered differences in social processing among autistic teens who pass as non-autistic. Researchers from Drexel University investigated how these teens respond to faces and emotional cues, revealing faster automatic responses and reduced emotional reactivity to subtle facial expressions. The focus was on understanding social masking, where autistic teens hide their diagnosis in social settings, often to appear non-autistic. The study included community-recruited participants, some without formal autism diagnoses, broadening the scope of understanding about the autistic population. Teens were classified based on comparisons between diagnostic assessments and classroom observations by teachers and parents. Nearly half, 44%, of the community-recruited autistic teens passed as non-autistic in classroom settings, often going unnoticed. The research is part of a larger National Institute of Mental Health-funded project examining social outcomes in autistic youth. It highlights that masking involves specific brain activity differences, as shown through event-related potentials related to facial recognition. Lead author Matthew Lerner emphasized that these findings shed light on the mental effort and potential psychological toll of social masking. The study also provides new evidence of how masking alters social information processing in the brain, distinct from both non-autistic peers and non-masking autistic teens. This has been your daily brief in science. To read more about these stories, follow the links in the episode bio. You can also subscribe to these updates via email at www.brief.news. For more daily podcasts about the topics you love, visit brief.news/podcasts. Tune in tomorrow. We'll be back with everything you need to know.